Thank you for inviting me to talk at the seminar, especially being in such a STEAM company who are talking, and then there's me. But hopefully I can show you some examples of, if you've taken everything into consideration, what we've learned today, and even disregarded a bit of it, you can still do some incredible, very exciting cognitive neuroscience with this data. So originally the plan was I was going to talk about two areas of things which are not the cortex. So I was going to talk about some deep sources, primarily the hippocampus, and then I was going to talk about the spine. I will ditch the spine stuff for now, but that's not the bad thing, because I'm going to be talking about this on Tuesday. So as a forward sell, come to my talk on this. Okay, so I'd like to give at least a theoretical case of why OPMs might be useful for deep source imaging. Right, so you've seen this plot already. Larry gave a version of this, and I want to particularly focus on the theoretical advantage of the OPMs. By bringing the sensors a lot closer to the head, you inherently boost the signal you can record. And in these deeper areas, where some of these structures may exist, you get a boost. Obviously, at the moment, with the current designs of the OPMs, the noise more than cancels this out. So the word theoretical here is doing a lot of the heavy lifting. Let me put laser pen on. But you can get around this because of the modular form factor of an OPM system. You can build it however you want. So in London, what we do is we build for a person 3D printed helmets. So we have essentially perfect co-registration between the sources and the anatomy within tolerances of the manufacturer. And because it's such a lightweight helmet, you can wear one of these for hours. So if you can't get it up through SNR through just cleaner data, you can just do these experiments for longer and get to these deeper structures. So what we've been looking at UCL is primarily the hippocampus, because lots of people down there are very, very interested in this. And for the non-neurosciencey people in the room, myself included, it's these red fellas here in this animation, so they're quite deep in the brain. In terms of some of the things they're useful for, they allow you to consolidate your short-term information into long-term memory. A lot of it gets routed through there. And there's lots of attributes to navigation which go through the hippocampus and some nearby local structures. Now, you might think this is a deep source, and lots of reviewers do tell us this, is that you can't see the hippocampuses with an MEG system, let alone an OPM. And I would say that's not true. So the CA1 and CA3 areas of the hippocampus, which is where a lot of mainly at the front of the hippocampus, the anterior areas, they have a very similar morphology to the cortex. So they have these pyramidal cells, a bit like our layer 5 cells, which are perpendicular to the folding of the hippocampus, which means that we can use similar generative models that we do for the cortex. So importantly, they produce open fields which can be detected. And secondly, there are way more pyramidal cells per square millimeter in the hippocampus than there are anywhere in the cortex. So we've got a good fighting chance of being able to image function from this area. So let's talk about the first hippocampal OPM experiment done at UCL. Or actually, I think technically this was recorded in Nottingham, but yeah. So we did this way back in about 2018, which is actually so long ago. It was actually before I joined any OPM projects. So they just wanted to see if we could just image hippocampal function doing quite a standard cognitive experiment to make the hippocampus light up. So you can see we've got one of the early generations of the 3D printed helmets here. These were much heavier. We've made them lighter since then, and there's roughly 20 or 30 sensors located to the dorsal areas and the side areas of the head, because that's more likely where we thought we were able to pick up the hippocampus. So it's designed specifically to get those areas as well as possible. Okay, so they were asked to imagine a scene during an experiment, and then in counter trials, they were also asked to just count backwards in their heads. And if you try and contrast what's the 4 to 8 hertz power, which is the theta band, during those two epochs, what you get is some activation. In these 3 subjects, it was all the way over the brain. Because it was such an early study, we only have 3 people, so we did t-testing within each person individually across their trials, and then we produced a conjunction map, which gave us a really nice localization here, roughly in the right anterior hippocampus. So far, so good. But obviously, you've got to, in the early days of this, you had to be able to show that you could do this in a conventional MEG system. And that's precisely what happened. But as you might notice from the SquidMeg results using the same pipeline, the same experiment, the activation flips into the other hemisphere, which is an interesting thing to happen. So I guess a question is why? And it gets even more baffling if you look at an fMRI experiment of this, because also you fly all the way to Australia and realize there's a typo in your slides. So what we did was we used LCMV beamforming, which by its nature assumes every source in the brain is independent of each other. And if it thinks there's correlated sources, if left untreated, it will try and suppress that data because it thinks it's interference, for example. And as you can see here, we did some Bayesian modeling to have a source inversion technique, which was a bit like a beamformer. And then we allowed it to be able to see and unsee correlated sources. And these areas in red represent where, if you try and use this treatment, it still can't tell the difference between two correlated sources and two uncorrelated sources in those areas. So we have the strong belief that even though the hippocampus they're quite large structures, the anterior areas are just so deep, even with OPMs, especially if you've got poor coverage, that you still have these areas mixing. So you've got to be very careful. So if you've only got a few sensors, you've got to be really creative with placement. So sometimes you should do a bit of like electromagnetic modeling to work out where would be the best place to put these sensors. And this is something which Tim Tierney did a while ago. And what he realized was that if you look at the forward models for hippocampal sources, you get three hotspots. So you get one on the side of the head, one on the other side of the head. But interestingly, there should be one roughly by the roof of the mouth, mainly because of its proximity to the hippocampuses. So if you can somehow get a sensor near the mouth, then in theory, you might be able to pick up a bit more signal. And this might be super useful if you've only got one or two sensors. It may be the best ways to go through your mouth. So this is something we tried. We've only ever tried this once, unsurprisingly. But what we did was we made a dental brace, put in a first-gen Q-spin sensor, and then we got our participants to bite down on this. I suspect this is not going to catch on, but it certainly makes for a resting imagery. The good news is in some ways it wasn't in vain at all, because if you do a sensor level t-tests on the activations, this sensor in the mouth was the one which gave the largest magnitude change in t-value. What we actually also found out, which I've hidden from this slide, is that if you took that sensor away from the source analysis, it made very little difference in the end. But if you've got such sparse coverage that you can't do proper source reconstruction, this creative use of sensors is completely valid. And just to check, it was definitely picking up something from the hippocampus. Tim did a DICS coherence image of that sensor versus all the other sensor source reconstructed activity. And the largest coherence was in this case, the right hippocampus. Because DICS also works a bit like a beamformer, we've only got one hemisphere, so that wasn't too much of a surprise. But actually, you can just replicate MEG experiments like you would in the squid, but then if you've got this system which is completely lightweight and completely modular, why would you want to keep still? And I want to give a neuroscientific reason why this might be a bad thing to just sit still and do small experiments like this. So this is a paper which came out in Communications Biology last winter from Berlin. They got people to do a human version of the Morris water maze, where you remember things in a virtual environment and you walk up to them and you pick them up. And then later on, we remove all the items from the environment and we tell you to go and stand where you thought that item was. And they did this in two ways. One with a kind of a sit-down setup where you use the joypad to explore a 3D environment, and one where they did this in VR, where they could walk around freely in the room. And they did this with healthy controls, and they did this with people who had hippocampal lesions in one of their hemispheres. And what was really striking is that for those who have in blue, which were these people who had temporal lobe resections because of the lesions, their performance in doing this multi-sensory version of the experiment where they got to walk and move, they performed far better, which is basically saying the hippocampus alone is only doing so much of this. And you need to think of the hippocampus as a smaller part of a much, much larger network. So your experiments really have to go in this direction to get a true understanding of what's going on. So we're trying to get to the early steps of doing this, of doing these more complex experiments. So this is a talk. This will be going into more detail by Kasia on Wednesday or Thursday. She's got to talk about this, where she's been trying to disassociate different elements of navigation, so the egocentric and the allocentric. So one's about how you perceive the world, and one's more of a map-like navigation around the world, where she got people to face the room in different directions and move a bit, and then had to imagine movement. And as a sanity check, she got people to imagine standing still and have their eyes shut, and also imagine moving around the room. And incredibly, this took very little effort to get this image, I have been told. Just by contrasting theta power between these standing still and imagining movement, lit up the hippocampus. So even with the fact that people are standing and there's very little active shielding in this experiment, just by having good head coverage and a good handle on modeling the noise, means like these deep sources just light up, which is really, really cool. And I've tried to keep this super, super short. So one final thing of some very exciting stuff, which is coming out of the lab, is we're doing these experiments where if you want to get people to remember new novel environments, you must first create an entire universe where there's no possibility they've ever seen it before. And this is what's been happening in Eleanor Maguire's lab. So Nick Alexander and Rob Seymour have been mainly leading this project, where they've built an entire virtual town and you have to walk around the town. So if you have a look, you can see he's got these retroreflective markers on his feet. You can't explore the town unless you're walking on the spot. So in theory, you can go on this journey with a virtual avatar, going to the zoo, watching a band play in the park. And this town is massive. It roughly works out to about 1 square kilometer of virtual environment you explore, which, if any of you have ever played Call of Duty Warzone, that's about 1/5 the size of the map you play on. This thing is massive. And presumably the results are also going to be equally as massive as well. I would like to show you them, but it's best that you read the paper when it comes out, and there should be a preprint imminently. So I'm going to stop there to let Sarang talk. So I just want to thank everybody who's taken part in this part. You can see us all on Tuesday, it's fine. But yeah, I just want to thank everyone who's got us to this point at UCL. This is a cast of several million, and also I realized I've left off the slides, Vishal, for his help over the years of helping us just do mad things with his OPMs. So thank you.